Thank you very much, Petteri, for this nice presentation. Even as an atmospheric scientist, I know something about diseases. I suggest we go with Emerson Del Ponte as an independent examiner first. You can take whatever 10 to 15 minutes to ask questions, and then we go for a further round if required.
Okay, thank you very much. I'd like to first thank you, Petteri, for the invitation and also the committee, Professor Bruce and Professor Alexei. It's really a pleasure to be here, coming from a long and far away country. It's a long trip, and I'm fine, I'm feeling well. Yes, so you are here you are showing your working that you have done during 3 years, right? This is 3 years of
3
work.
years, yes.
3 years, and you have 4 chapters.
Mm-hmm.
So you were able to put out 4 chapters within 3 years, which is really great. It's really, when I was reading your work, I could see a lot of work. So you worked very hard. But obviously, this is not an independent work. You have help from the team.
Yes.
This is really nice that you have the teamwork. So many people helping you, which is good. And you had a mix of work from the DNA up to the field. You could learn a lot of different concepts, different tools and approaches, going from the lab, from the bench, going to the greenhouse and potted plants and into the field.
Yes.
So this gave you a really nice overview and broad techniques, and you learned a lot of concepts about epidemiology. So this is really a very classic epidemiology work, which is really good. I like it very much. So, but within 3 years, and you've been so busy doing work, I wonder how many epidemiology books you've read during this time. Because I know that you don't have classes here, so you did not attend any epidemiology course formally, but you got access to textbooks, articles, and to the literature, which is important for you to base your planning, research, and all your work is based upon what has been done before you, right?
Yes.
And I have some questions about some concepts. Okay, when I was reading as an epidemiologist myself, I like to check if you know well some of these concepts, which is really important for you as a doctor in epidemiology. So everybody will look at your thesis and say, wow, this guy is an epidemiologist, because the kind of work you did allows you to say, wow, I did a lot of phase epidemiology work, and not only experimental, but you did a little bit of modeling too. Okay. When you mentioned about the host damage, is there any other term in the literature that refers to the host damage that's more intuitive and more, I think it's a better term?
Severity?
Yes, but when you mean host damage, is that the damage can be visible damage, invisible damage. The damage is quite broad, it doesn't say much. Is there any other term that you could use instead of host damage and pathogen reproduction? Because you assessed something that was visible.
Yes.
Right? Not only for our eyes, but for the camera.
Yes.
So you could pick colors and stuff. And is there any other term?
Besides severity, I don't think I'm aware of any term for that would be somehow similar.
And have you heard of symptoms? Symptoms.
Symptoms, yes.
Yeah, that's what we see.
Yeah.
And the host damage, I think what you measured, what you quantified was the symptoms.
Yeah, sure.
Yeah, symptoms. And in pathology, we say that when we see something on top of those symptoms, we call signs, which is the reproduction part of the pathogen. And it's more intuitive. And there's another term that you have used to refer to this amount of these symptoms on the leaf. You call it PLACL. So this is an acronym.
Yeah, but
this is based on the previous paper, right?
Yeah.
So, but there is another term that explains that acronym, right? And you just mentioned that.
You mean severity?
Yes.
Yes.
Yeah, that's more. It can be a kind of vague term because people use for different things like the index, and they use this across different contexts. And I think you define it well, but that's very intuitive, and we have a proportion matter fact that it sees.
Yes.
And the symptoms that are visible.
So when we were writing the first paper, we were thinking of this terminology, and of course we realized that severity is a much more common word for the host damage, for the PLACL. But then sometimes severity is also defined based on percentage of leaf area covered by good media bearing lesions or something like that, based on the good media. And we thought that we could refer to host damage and also the pathogen reproduction with severity. And we wanted to make clear distinction so we don't use this perhaps more intuitive but probably less precisely defined terminology.
Yeah, I see that you try to find a more specialized term to facilitate the communication between people working on this disease, to make clear that you were referring to symptoms and signs and things like that. And when you mention your title is quite broad, it's measuring and controlling, right? Measuring and controlling the pathogen, right? When we mention epidemiology, and is it epidemiology of the disease or the pathogen? You control the disease or you control the pathogen?
I would say we control the pathogen.
Right. How you control the pathogen?
Well, of course, it's based on the disease symptoms, and we want to suppress the disease. But well, I always think of it as a kind of management of the pathogen population. We want to suppress or somehow manage the pathogen population. Sometimes we probably don't want to suppress too much, or not creating so much selection pressure for the pathogen.
But you measure it to make sure that you control the pathogen you measure the disease.
Yeah, we measure the disease. Yes,
the disease.
That's right.
And all the processes, they depend on the host too. So if you use a cultivar that suppresses the pathogen, you're not actually killing the pathogen like using a fungicide. Then you control the pathogen.
Yeah, that's true.
Yeah.
That's true.
But I think the most correct here would say that epidemiology refers to the disease
of the
processes, and then you can control the pathogen. But I think it's more in our field we say we control the disease. So we can suppress disease, or depending on the kind of pathogen life cycle, you can apply something that's is targeting the pathogen or the plant.
Yes.
Usually our target is the plant. You protect the plant.
Yes.
You try to modify the plant.
Yeah. And of course the eventual goal is to reduce the disease.
When you say measuring, there's one section in your text you say you measure the quantitative resistance. Actually, you measure.
We measure
the quantitative resistance, or
we measure severity of the disease.
You
and
quantify.
Yes, we quantify the disease levels, and so then we use that as a proxy of the quantitative resistance. So if we see lower severity of the disease, then we assume that it's
okay. You are measuring the traits that are associated with the resistance.
Yeah.
Just to make sure these terms are okay or not.
Yeah, it was
you
also
gotta read carefully. When you read carefully your work, I could take a lot of these inconsistencies with the previous literature.
Yeah, we had discussion with the reviewers about the use of component of resistance, for example.
Okay.
You want us to be really careful for not to refer too directly to the resistance when we are actually measuring the disease.
And actually, you are at the edge of a field that there's a we have a plant pathologists in one side, we have the breeders in the other side. So there is a jargon that is specific to each of these groups. So then eventually there's a collision. You don't agree. And yeah, and you need to make sure you use something that is understandable by both groups. Okay. But it's still, you have the literature to give you feedback or background on this. Okay, I'd like to talk a little bit about chapter 1. And you had this high number of cultivars, more than 300, right? Fields. They were planted at the same time.
Yes.
So are they synchronous in terms of phenology?
No, they are not perfectly synchronous. And we have some data about flowering, but we didn't include that in the analysis of the.
And they probably have different shapes and heights.
Yes,
and,
they have different shapes and heights.
Size of
yeah,
leaves.
Leaf size and the distance between leaves, and they are of course timing.
And do you think this could be a factor affecting the results?
Yeah, sure. For example, if we have some higher cultivars that manage to escape the disease, the leaves grow, trees emerge and the stem elongate so much that the healthy leaves are kind of away from the disease source.
But you could not account for this fact.
No,
because you were just sampling leaves within a certain plot at the same time. You
yes.
sampled over 2 times.
Yes.
It was before flowering and after flowering.
Yes.
Right? About 1 month.
1 month, a little bit more, 5 weeks.
So we did not have more precise assessments over time, just visually. Visually you had 3 assessments. Visually, yes. And then using the image analysis, you sampled over 2 times.
Yes, 2 times.
And you sampled a few leaves. How many leaves you obtained from each plot? About
so
45 leaves?
From each plot, at each time point, we got about 16 leaves. We had 2 replicates. So
16?
Yes.
And then you had another...
Yeah, we had 2 complete replicates, so we got around 32 leaves for each time point, and then 2 time points, about 60 leaves in total.
These replicates you planted at the same time?
Yes,
also.
yes. They were something like 100 meters.
Block.
Yes.
Yeah.
Yes.
Okay, what is the trade-off of sampling fewer leaves, let's say, fewer leaves, and get a great amount of data from fewer leaves instead of sampling more leaves and get a more rough measurement of the disease in your case?
Yeah, clearly there is strong trade-off. If we collect really really large samples, then we can't characterize them so precisely. And for example, one of the weaknesses of the work in the first chapter was that we were not able to characterize the disease levels, the overall disease levels in the plots, because we were collecting infected leaves. Okay. So we don't know exactly which proportion of the leaves were infected. We don't know the incidence of the disease in those plots.
Yes, so you could not really track the epidemics over time using this method. You just you sampled during 2 different times, and you could see differences in terms of the disease levels. So first time you have less disease, and then you have more.
Yes, yeah, and those were also
and
different
you could predict
layers. So
the second time based on the level that you could see the first time, and then you could use that information to predict. Okay. But all the epidemics, they were in the same area, in the same environment.
Yes.
I was a little bit curious. I have a question first about this new index. You have this PLACL, and you have the number of picnidia per lesion, right?
Yes.
It's per size of lesion or per
yes,
lesion?
per unit area of lesion.
Per unit area of lesion area.
Lesion area, yes.
And the index was confusing about how you calculate it, because in the manuscript it's not clear to me how you calculate. I tried to calculate using some of those numbers. So it's the number of picnidia per leaf, the total leaf, or you multiply something like.
You mean how we calculate the picnidia density?
Yeah.
So that's
the picnidia density for the leaf.
Yes. So we have total number of picnidia for the leaf, and we have total amount of damaged leaf tissue or diseased leaf area, and density comes from that, dividing the total number of criteria on that leaf by
the total area with the symptoms.
yeah, total area of symptoms.
Yeah.
And
So
then
it's not. So what's the difference when you first you calculate the number of meters per unit area of the lesion?
well, that's the final measure. But what the macro actually does is it just measures the symptomatic area and the total leaf area, and it counts criteria. And then this criteria density we got by dividing the total number of criteria on the leaf by the symptomatic area on the.
So this gives us the raw leaf that you have, whole leaf.
Raw lesion. If we divide the total criteria number
yes.
with symptomatic area, then it's criteria density in lesions. And then if we divide it by the total leaf area, then it's the criteria density.
Okay, it's a kind of index. You multiply the first measure by the other measure, right? It's a kind of index. Yeah, it was a little bit confusing to me because you used this acronym, the PLC, and then you have the row leaf, and the row leaf was a little bit complicated to understand how you got to those measurements. And you could have just simply used the percent area affected by the lesions and the number of pit images per lesion and the index, criteria index. Because you actually have an index. When you multiply two different things, you have an index. But when you assess it using both of these measures, let's say these variables that you got, you could not differentiate much because there was a large block of cultivars.
Yes.
You could not.
Most of the cultivars were in the
have some
medium
on the top
range.
and some at the bottom.
Yeah, so we had most of the differences in the end of the distribution. And actually here the colors, different colors reflect the statistical differences between cultivars that we were able to find. So all these orange dots here reflect cultivars that are not different from each other, but they are different from the differential color, so the two extremes.
And do you know why you could not tell them apart? Do you know why? That's all the background that you have, all those points are it's the variation that you have.
Yeah, it's individual.
Across the leaves.
Yes.
You have a lot of variation.
Yes.
Even using this very precise, maybe because you use a very precise method, you could make this variation that maybe using the visual symptoms you could not get. So this variation is so large. And so you can see that for some cultivars in the middle, you can have a very low number of media, high number, or the area factors can be very different.
Yeah, it's really variable within each cultivar. And I think this shape really reflects the traits of the cultivars that we had. So most of the cultivars are kind of average or performing moderately well. Only some of them are exceptionally good, so that we can tell them apart from the rest, and also some are exceptionally bad. And actually, when we did similar plots or rankings with the visual score, we were even less able to tell them apart. And that's likely because those fields were treated by fungicides. So even though it was really bad year for the farmers, so it was really high levels of septorium fields generally, in our study the levels were kind of moderate, and probably that affects our ability to really tell them apart. Probably without fungicides we would get larger differences or more pronounced differences. But then we would most likely have also other diseases that might happen.
I wonder if you got any information, previous information about those cultivars according to the resistance level, and then you could check if using this method they would group the same. They would group the same as the previous.
In
I don't know.
the
Do you have
previous
this
studies.
information about the cultivars?
Yeah. So Some of these cultivars were used in smaller field experiment in previous year, and there was significant correlation between the rankings, but it was kind of moderate or moderately weak correlation. And when I spoke with some breeders, they thought that our correlations are actually really high in their view, and they say that it's most likely because it's so difficult to tell the cultivars apart. So if we try to get correlation between two rankings, most of the cultivars are in the middle range. So it's really easy to get just by random large differences in the ranking.
You have only very few groups, like 3 or 4.
Yes. Yes.
When you use the visual assessment, you split more into groups, or
no? In the visual scoring, we were not even able to split. So I can show you one of the figures in the. So here are the AUDPC values from plots, and these individual dots reflect the basically contribution of each visual assessment to the AUDPC. So they reflect the disease levels on those plots. And as we see here, all of the most of the cultivars has really similar scoring for this time point, and also here there were not so many differences. And on 1 to 9 scale in visual scoring, we had I think highest score was 4 overall. So there were not really high levels of disease.
Okay, one question more about this chapter 1. Did you recommend this method, the image analysis tool? Is it ready to be used by breeders, by the pharmacologists, or is there something that you just use this method to learn more about what to look for in the future? So visual assessments can be improved.
Mm-hmm.
Did you think about this? Because when I was reading, it was not clear to me what the take-home method was. If you're really proposing that people use this it's very complicated. You take all the photos and you present the images. There is the macro, but still it's a lot of work.
Yes.
To get to the image.
Yeah.
So what is there's something I was just wondering if the visual assessments could improve just because you know better what to look for now. If you use a method that is very precise. So, did you think of any standard area diagrams that you could build just using this database of images that maybe people could use to rate the score faster?
Yeah, that might be a really good idea for individual lead scoring, and in our measurements, the measurements based on good media numbers were more robust against the temporal or spatial variation. So I think it would really make sense to try to account for good media in the visual scoring. But then, of course, it's more subjective nature of accounting for the good media density. And I think here there is kind of similar trade-off, as you said before, between high intensity and high precision of measurements, but smaller sample size, and then large sample size with lower intensity of measurements. So with the image analysis, it really requires some work, and I think really well-trained plant pathologists can probably deliver as reliable results with visual scoring much faster. But I think the main audience for this or target group for this method would be academic plant pathologists. All the breeders that I have spoken with say that it's not possible. They have millions of breeding plants. They are not going to collect individually from all of the plots. Just go to the feed and look at 1 and 3 like this. So but all the method is open source. It's free, and source code is available, and we are trying to develop it further. And I know that several plant pathology groups are using it in Europe and I think also in Australia.
this screening work is done out in the field, or they use potted plants in the greenhouse to screen for this pathogen, for this screening genotypes is done in the field?
In the breeding.
Yeah, breeding.
In field.
In the field.
Yes.
Yeah. I was just wondering how maybe you can have some good information about, maybe you can improve this image analysis too. Now that you know where to look at,
yes.
maybe you can like have something like taking photos, or like small photos from
of the plots.
higher number of leaves across different heights, and then you have this precise area, and then you take a lot of images, and then you process those images instead of taking the image from the whole leaf.
Yeah.
You just
We
go to
do-
the field, use your phone, and you can
Yes.
take very precise measurements.
Yeah, we know there are a couple of companies that are developing mobile device applications for at least detecting some diseases, and we also tried that, and we also took some steps towards the indoor plot measurement that you mentioned. So we had photos of the canopy, and we tried to see if we can visually detect lesions, and we think that if we can count lesions or somehow detect them visually, then probably machine can do it also. But we were not able to take that really far. But in one more promising line, we collaborate with crop science people, and they used our leaf level data and their hyperspectral measurement data. So they had a camera of the field, and they took hyperspectral images. And there are some promises for faster automated pixel measurement.
Just one final one, just missed. You did not connect.
Then we should move on.
Yeah.
Now you're at good time. So this is your last one.
The last one, okay. You did not have new data from this spot. Did you get new data?
No.
Couldn't that be important to have
Yes.
new data, because you are trying to say what kind of variables is most important, but really for a farmer, he would like to have a good crop price.
Yes.
And it's still that
Yield matters, yes. So here we need to rely on some previous work that show that disease levels on black leaf and black mouse one leaves are important for the yield production. But the perfect case here would be to have a complete box of the cultivars growing in disease-free conditions to be able to differentiate between this yield without the disease and yield with the disease.
Okay, I'm fine now. I'm done. Thank you very much.
We can always come back.
Yeah,
Give another
sure.
shot at it. Sure. But we should move. Give
No, it's fine.
Alexi a turn here. He doesn't get any rest, but you can keep it going here.
So thank you very much, Väitöli, for your talk, and I have to say that I feel very lucky to be able to work with you during the last 3 years and to advise your PhD research. Great job, very well done. So I have several questions about kind of focusing on a bigger picture of the population biology of this pathogen, in particular from the evolutionary perspective, because I know that you'll be working more with evolution in the nearest future. So experts on this pathogen often say that the population is very diverse genetically. So the first question is, do you think this genetic diversity also translates into high phenotypic diversity of different strains?
I think so. Yes. So when we measure any phenotype of the pathogen, either in plant or in vitro, we always see lots of differences between strains, be it function resistance or latent period or lesion size or critical production on different cultivars. And then of course it depends on the conditions.
Right. Then to follow up on that, this is a pathogen of an important crop, wheat, and people are trying to control the disease. and in this way, they are also suppressing the pathogen population. So we think that the population is under strong directional selection, and kind of what evolutionary theory tells us that if a population is under strong directional selection, the diversity should decrease over time. But that's not really what we see. How is this population able to maintain this extremely high levels of genetic diversity and corresponding phenotypic diversity?
Yeah, so I think the case here is that we are not really able to suppress the pathogen population significantly. So there are always fields where the pathogen can survive very well, and even in best fields of resistant crop treated with fungicide, we have some levels of disease. And there are also, for example, in fungicide spraying, not all the leaves are perfectly covered by the fungicide. And in the bottom of the canopy, on dead leaves, the host resistance is not active anymore, and the pathogen can survive there. And then when it's having sexual reproduction every year, it's mixing basically all the material that's around. And if I remember correctly, one estimate was that 90% of global genetic variation is present in just a single field when we sample intensive measure the diversity. So we can't suppress the diversity.
So okay, you said that we cannot suppress sufficiently the pathogen population, but how does this inability to suppress it, how does it allow the maintenance of the diversity?
Well, there is if you think about the directional selection, I think when what I mean with not suppressing the pathogen is that we are not really imposing the directional selection on entire population. Only part of the population is affected by the selection, and large part of the population is not under the selection, and that's one way of maintaining it.
But okay, you can have a large part of the population, but why should it become diverse? I mean, what are the mechanisms that
because of the sexual reproduction, the genetic material from different strains from the part of the population that is under the strong selection and from the part of the population that's not under the selection, they mix, and then for example, if we are selecting for really fungicide resistant strains on fungicide treated leaves, we might get some reduction of diversity there or strong directional selection. But any strain can actually land on that lesion and mate with the strain, which means that the strain that was not under selection can mate with the selected strain and kind of break the genetic combinations. We can't continuously keep same strains under selection, except in greenhouse.
Okay. So then a bit more specific questions on the chapters. So related to what Emerson was asking. So what we see, if we could show the previous figure. So what we see is a very high, like the outcomes within each cultivars. There's a very high variation.
Yes.
As Emerson pointed out. So what do you think are the sources, the main sources of this variation? And on top of that, how, if you would be able to follow up on this experiment, what would be a way to disentangle and tease apart these different sources of variation within each cultivar?
Within each cultivar, yes. So I think there are some stochasticity, of course, where the spores land and which leaves happen to be in favorable microclimate or things like that. Then in the fungicide spraying there can be heterogeneity. Some leaves might get more fungicide and some leaves don't. And what else I have in mind? Well, something that we observed in the second chapter was that when we inoculate individual cultivar with individual strain. So genetic material is the same. We keep them in same environment in the greenhouse. We see some lesions appearing after 2 weeks, and some lesions appearing only after 5 weeks. So there's lots of unknown variation or variation that's not known where it comes. When all the genetics and environment is the same, there's a lot of difference in the development of disease. And how to account for different sources? Well, I think with the fungicide, it might be possible to have fungicide treated and non-treated plots to see if that affects it. And then probably when we were sampling, we should define better. Probably train the people collecting the leaves better, so that they are not by accident collecting naturally senescent leaves or leaves from the borders of the plot and things like that, to try to keep the sample quality as good as possible.
But so, I mean, related to my previous question about the diversity. So, and this is a natural infection. So what?
So one part of the variation is as the stochasticity in the spore landing where the inoculum is present.
Yes.
But also when the inoculum is present, which genotypes, which strains are present there. Some strains might be much more aggressive. If we get just a single lesion of aggressive strain in leaf, it creates a pypidia, and then those spores will spread over that leaf really easily, and then that particular leaf might be much more diseased than others. And then of course we can account for that by measuring the genetic diversity or observing some isolates on the leaves.
Yeah. Okay. Perfect. Thank you. So. Now about the chapter 2 on the measurement of infection efficiency. So the measurements you conducted were kind of a proof of principle, and so you looked at infection efficiency and the latent periods in one particular pathogen strain and in one which genotype.
Yes.
So do you expect? I mean, partly you already answered that. So I think you would expect a lot of variation between different strains in these parameters. Yes. But then the question is, how many strains would you need to characterize in order to really kind of capture the characteristic distribution to characterize the whole population?
That's a good question. How many strains? I think that that might be. We might approach that with similar methods as with the study that I mentioned, where they find that 90% of global variation, genetic variation, is present in one field. So we might, if we had really large isolated collection, and then we know something about the global genetic variation, we might kind of try different sample sizes to see where we achieve some level of 80% or 90% or something like that of the global variation.
Mm-hmm.
Then of course we don't know if that genetic variation that we observe is really related to our phenotype, but I think that would be my guess.
Mm-hmm. So let's move on to the next chapter about the dispersal. So do you think this parameter of dispersal, the dispersal radius, is it a trait that's under selection in this population? And if so, what kind of selection?
It's hard to see how the pathogen could really affect the spread of the spores. I mean, there is some variation in the spore size, and but the spores are anyway really small compared to size of droplets of rain splash. So I think that that might not affect it. But one thing that pathogene can do is to make sure that the spores are available when spread is possible. So when rain event, when we have rain, and one way for that might be what we see in the heterogeneity of latent periods. Some lesions after 2 weeks they are ready, waiting for rain from 2 weeks to probably 3 weeks. I don't know when the spore will die, but then some fresh spores will appear later in 4 or 5 weeks.
You could get. Maybe you're one more question.
Okay. So now, one question about the last chapter on cultural mixtures. So, if you were able to repeat this experiment and have a large team doing any kind of measurements that you like, so what would be a way to distinguish the contributions of different mechanisms of disease suppression in cultural mixtures? For example, distinguishing between the disease suppression due to the density effect or dilution effect and the barrier effect. I mean, how would you go about doing that?
Well, if I had infinite resources, then it could be possible to plant poor stands of the cultivars in different densities to see how the density affects it. Then we will probably put some physical barriers, not plants, but or probably some other plants, but even better kind of artificial wheat plants. Mix them with the canopy.
Or non-host, maybe
probably non-host, like barley or something like that. And then one thing that would be really interesting would be what's the level of epidemic on different components of the mixture. And the French people got really nice work on that. I think it's not published yet, but they colored the seeds before sowing, so that when they go to the field to pick the seedlings, they were still able to see the seed and see if it's resistant also to cultivar. And then later on, the canopy structure was more or less similar, except that one cultivar was with awns and one was without. So after heading, they were again able to differentiate between resistant and susceptible cultivar.
Okay, thank you.
Okay. I'll try to keep it to under 20 minutes. So first of all, I just want to say I really liked your general conclusions chapter. I think it was very well put together. This is where you summarized all of your findings and sort of put them into an application sort of network. How would you apply these findings? And I think I'm tempted to make this mandatory reading for all of our PhD students, because I think you did such a nice summary of how this research translates into application. That was very nice. Thank you for that. And I want to start with a question on your 4th chapter, which was the mixture experiment. And I don't know if you're aware of the fact, when I was a PhD student more than 30 years ago, my PhD research was on mixture effects, and that was with barley plants and the barley scald pathogen. And what I found was that once you got to 33% resistance percentage in the mixture, you got a significant mixture effect. You got 25%. And the reason I say this is these mixture experiments go back at least 50 years, and they're deep and broad around the world, and they 95% of the time show a really profound positive influence on diseases, and a positive influence on yield, and a positive influence on yield stability. Everything.
Yes.
So why are we still growing clonal monocultures? What the scientific data is so strong and exhaustive. Why are we still doing clonal monocultures?
Yeah, I think there's bad gap. First of all, there's big gap from academic research to applications. I think it's quite common that when academic scientists looks at some problem like disease that we are looking at here, they see the problem, they study it, they find a solution. Then they publish the solution and they think the problem is solved. it's all only in their mind. But no one is using the solution. And when I presented the cultivar mixture work in the International Symposium of Ciri leaf blights a few weeks ago, after the talk, one breeder said I was asked about her opinion on the cultivar mixtures, and her opinion was it's kind of a nice idea, but it will never work. From her point of view.
Did she say why?
Well, I think her idea was that it will never work for the breeders. And I don't think even that is true. But with their current system of how they plant the millions of breeding lines, how they score for the traits and things like that, it would require really major changes in all their approaches. And then she was speaking about introducing mixtures. The mixture breeding aside of the conventional breeding, they would require double resources for everything and like that. But probably some smaller breeding company, like a young startup, might take up some ideas and use them. But I really see more potential in trying to convince farmers to use mixtures, because they can use mixtures. They can buy seeds and mix them themselves, and in quite many markets it's possible to sell mixed seed.
So if you get the farmers doing their own experiments and seeing positive outcomes, then they're more likely to adopt that technology.
Yes.
So really, we shouldn't be communicating to the breeding companies, we should be communicating directly to the farmers.
Yeah.
And once you figure out how to do that, please tell me. Okay, so now I'll go to chapter 2, and this wonderful number you came up with, which is 4%. So about 4% of the spores applied onto the leaves on average formed a lesion. Now that number is an interesting number. Do you know how well that number compares with similar experiments done with other pathogens?
With other pathogens, there was review on infection efficiency measurements. The methods are really variable, so the methodology is not directly comparable. In some cases, they get over 50% efficiencies, and in some cases, it's around 1 to 10%. So I think it fits well, but it's probably in the lower end of the spectrum. And in one experiment of Helen Foles and others, they also got as kind of side result. They got infection efficiency, and in their case, it was around 50%.
Wow. There's a big difference. Same organisms, greenhouse experiments in both cases, 4% versus 50%. Which result do you trust more?
Well, both of the results are with single cultivar, single strain, and single conditions. So I wouldn't make really strong conclusions based on either of those, especially when we have two really different results. Even guessing in the middle would be bad.
Yes.
So yeah, I would like to measure it a couple of times more.
So in science, when you get these types of dichotomous results, different research groups working with the same organism, asking the same question, maybe using different approaches, but they come to wildly different numbers, what's a good strategy for finding the truth? How would you propose to find the truth there?
So I think first of all we would like to look at the possible reasons of the different results. So starting from the strain and cultivar that we are using, we could measure those genetically identical combinations in different conditions. Then inoculation method might affect. In their case, they put droplet on leaf and then spread with finger, and we sprayed fog of the spore suspension, and that might affect the placement of the spores, their ability to penetrate, things like that. And then, of course, the greenhouse conditions. So first, I would like to try to unify the methods and conditions, and then try similar genetic material.
Surprisingly enough, I think that your findings are more likely to be correct, closer to the truth than their findings, where they rub the spores into the leaves with a finger, for example, introducing all kinds of wounds. But finally, I think it's important that people understand problems with all these methods and again, try to design something together. To get all the advantages of different approaches. Okay, one more question. And that's with the dispersal kernel experiment that you did. 99% of new infections fall within 1 meter of the source. How does that result compare with other experiments done by other people in other pathogens?
It compares very well. The mean dispersal is in the range of previous measurements starting from, I think, 70s already, where they measured dispersal of septorial nodorum or septorial TDC. Range pass dispersed pretty similar pathogens. The mean dispersal distance with range pass is usually around 20 centimeters or something like that. So it's pretty similar.
So it's really, really believable.
Yeah.
Okay, I think that's enough. Take it to.
Thank you. We're well past the hour, but if there's 1 or 2 more questions you would like to ask, then.
I have one.
Okay.
Think about this question, the previous one about the number of lesions is not so high in this disease, right? You could not see, so that the infection efficiency or infection rate, when you compare the number of viral spores and the number of lesions, you really have few number of lesions. You could not see, you have more these expanding lesions that are larger lesions instead of having a high number of small lesions. And you also notice that the latency period is quite long.
Yes.
Okay. You could not really, have you thought about the fact that maybe you have infections, you have successful infections, but it takes a lot of time, it takes a long time. So the lesions that appear start to grow faster, they go over the other lesions. Would there be a way that you could use another technique that you could really mark the initial infection of these lesions? Because when you use your method, it was vision. You were just counting the lesions over time.
Yes.
But that doesn't mean that you could not see the invisible ones. It takes a long time.
Yes.
So maybe there is a real, let's say, actual infection efficient that you cannot see, and that it's because this pathogen tends to the kind of damage that it does on the leaves just grows quickly and overshadow the other lesions. So just a thing I was having now when I said this. Maybe it explains because it's something that the disease is formed by lesions that are a little bit larger.
Yes.
And you provided data that about the latency period is very variable across the different lesions.
Yes.
Is there any technique that you?
So what we did was that we took photos every day on the leaves, because we often notice that we see young lesions, just the first yellow chlorotic spot in the leaf, and then another spot appearing probably 1 centimeter away from that, and 2 days later they would be merged. So by counting the lesion number every day, the newly appearing lesions, I believe we were able to get more precise number of total number of infections. But in the data we also noticed that there was saturation effect. So when we used different densities, we got this linear relationship with low density. So probably there the lesions were not overlapping too much. But with highest density, we got clear saturation, because the first set of lesions in 2 weeks was enough to cover the leaf, and then of course we don't see if anything is successful afterwards.
Yeah, but my question is there any method that you could track the invisible infection sites just before, because you track the visible symptoms?
Yeah, so
my idea is that there's something, there's a lot of infection sites that you cannot see. But when you see the symptom, it's overshadowing all the other infection sites that you could not account for.
there is actually some work done on the epiphytic growth of the the spores or the hyphae on leaf surface, and they measured the penetration success based on microscopy. So they were able to see when the spores actually penetrate. And I think in their case the penetration success was again higher than for us. So it might be that some of the visible penetrations, microscopically visible penetrations, do not lead to successful infection. And they also observed that some penetrations happened after 1 day, and some happened as late as 10 days after inoculation, and that's one factor affecting the variability in latent period. And then, of course, it's a question whether it's a true latent period that we see, whether we should start counting the latent period from inoculation or just from the penetration, how long it grows inside the leaf, and so on.
Do you have an idea? What would you
think?
Yeah, let's discuss that.
Yeah, maybe it's kind of a thermal imaging, kind of thermal imaging, some kind of different technique that's more precise that you can maybe track those infection sites, or maybe mark GFT something.
I was thinking GFT cleaner.
I don't know. It was just I don't have a solution in mind, but I think that maybe within hours that we could just track. But that would be the real efficiency. What we have here is the new, not infected, the new lesions over time.
Yeah, and I think it would be interesting. In this group, people developed a method for inoculating leaves with different fluorescent marker strains. So basically same strain with different markers. And then when spraying on the leaf, they get a different colored pygmedia. The spores coming out from pygmedia are of different color. And that might be used for detecting the mixed lesions. If we see that the individual lesion is bearing multiple colors, then it means that the single lesion is coming from several infection events.
The children are trying to rescue Dad.
He's getting
very strong signals.
He's getting outside help. I'm not sure this is.
I've got lots of outside help, I have to say.
Any other questions? Urgent questions from the committee? Questions from the audience? Maybe one or so? Everybody happy? Yes?
Yes,
please.
You had the dispersal panels experiment, and the answer was like 1 meter.
Yes.
But then you also mentioned that 90% of these strains are in any field anywhere in the world, which suggests that they're actually really quite good at going global.
Yes.
Is there a discrepancy? And what's the solution?
Well, what we measured was race class dispersal of asexual spores. But the sexual spores are wind dispersed, and they can fly over kilometers or hundreds of kilometers, and that leads to mixing of the genetic material.
So if that's the case, then does it really help that we have this one stage of very short dispersal? I mean, you're kind of like implying that the short dispersal can be used as a control mechanism by even manipulating.
Yes, the latent period for asexual reproduction is much shorter than for sexual. So there can be up to 6 cycles of asexual reproduction and 1 or probably 2 cycles of sexual reproduction in the season. And if we can suppress the asexual part, then we will stop monitor.
Last one.
There's another question over there. Would you give time?
In your field experiment in Tunisia, you did try to have the 2 resistant cultivars together in association. I don't think you reported on whether it did anything interesting.
I didn't show it, and it didn't do anything interesting.
Thank you.
Okay. Thank you very much for this interesting discussion. We will close the official public part of the defense now, and the committee should stay. We need to discuss for a few minutes, and the rest of you, thank you very much for coming. And don't go too far. We'll be back in a few minutes. Thank you very much. Thank you.